Hello everyone, this is Ashfaq Ahmed and I'm working in the field of bioinformatics and structural biology. So today we will talk about biology of regeneration and bioinformatics. I'm making this video on the request of a person, Mr. Abu Bakr Raja, and he asked me to make this kind of video that how we can do some work or do some research work or make a project using bioinformatics research tools. So it will be a shorter video, most probably of less than 15 minutes. So let's start it. So in first slide, uh, it's just an introduction of regeneration, that what regeneration actually is. Regeneration means that when the part or a complete organ regrow in adults human or regenerate in adults human, so we call it regeneration, such as it happens in liver also. If it is lost by some diseases or an injury, so the human body has the ability to regrow some of its cells and some of the portions, so we call it regeneration, or this field is called regenerative biology. So uh, today I'm going to show you a database which has been released in 2022, and I will also teach you how you can make your project using that database and some other bioinformatics research tools. So let's start it. This is the database. Its name is Regeneration Roadmap, or CNCB NGDC. I will share this link in the description section of this video, so you can take this link from there as well, or you can go through Google and search it, and it's the database of regenerative science. So the first thing is, how will you find information? So you need to click on the home if you are not on the home. Now here, if you go down, if you scroll down, okay, so just click here, regeneration related genes. Now you can see here bones, cartilage, endothelia, epithelia, and skin, spinal cord, stem cells, thymus, tooth, etc., etc. It's a longer list you can see here. So now you have to decide which organ you like or in which organ you are interested. For example, I am interested in, let's say, liver. Okay, so when you click this liver, you can see here that the system has given me some information. You can see here, and these information on per page you can select, like let's say I want 100 genes on per page, so you can see here. Now if you change your selection from skin, let's say from liver, let's say you change your selection to bone, so now the list changes to this one, and if you change your selection to heart, the list changes to this one. So we will select anything. Let's say I select a liver. That's okay. My main purpose is to teach you. Okay, so from here you need to find out your gene, or you need to select your gene. If you need a research paper for that gene involving in a particular regenerative process, then you can find it here. Just click here on this DUI, and it will goes to research paper you can see here. So here is your research paper, and you can download and read it about your gene. And these are the species. It may not only contain humans, it may also contain some other organism like molluscus or rabbits, anything else. So but if you are interested in humans, so just confirm the gene. This is the gene name in which tissue. So of course we clicked here liver, so it will be in liver. What is the protein name? So angioprotein 1 is located on chromosome 8 in the human, and start position in base pair is this is the end position, and total exon is R10, and this is the reference paper. The same way, this is another gene, and you can go down in this list. So the first thing, you can select your gene based on the major selection, then select an individual gene in that major selection, read papers, the initial paper from here, and then you can download other paper from PubMeds and research articles and make yourself a little bit familiar with that gene. That's it. Now, what this database can help you? This database can help you in 5 major areas. It will not be wrong if I say that these are the major areas that this database will help you. For example, it will help you transcriptomics, single cell transcriptomics, epigenomics, pharmacogenomics, and metabolomics. Still, after this video, if you need some more help using this database, then of course you need to click this help, and you will learn much more information that how to use this database. So I'm giving you a quick demo how we will use this database. So for example, we are selecting this gene ANGPT1. Remember its name, ANGPT1. So let's say I'm searching. You can also okay, I'm clicking on the transcriptomics, so I clicked here. Then from here, all you can say human and ANGPT1. It is selected here. Now you just click on search, and you will have some data here. Now this is the whole data. Only 2 data we have for this protein, but in your case maybe you have much more data. Then I would suggest to tweak this information. The p-value is actually the significance. So it is believed that a p-value smaller than 0.05 is significant. So both I have selected this, and we have again those 2 data set. It means they are significant. If you are interested in specific cells or tissue, then we have this option in case of liver or lungs, airways, epithelial cells or lungs, or you just all. So for different genes you will have much more options here. The log fold change means the transcriptomic level. If this value is in positive, it means upregulation. If this value is in negative, it means down regulation. If you see here in this protein, in these cellular tissues, in lungs, in Homo sapiens, and it's about the treatment scenario, and the sample was taken from the wounds, and this gene was down regulated in both these cases. Here is 1.3 minus 1.3, and here it's this. And if you want to read this paper, so these are the DOIs, just click it and you will find out more, and you can also download this information. Okay, so now further, let's say I'm going to explore. Okay, this is the RNA sequence data. Now if I'm interested in the gene ontology, we also call it GO, just click it here. We are still in the transcriptomic section. So the gene ontology basically tells you about the biological process, cellular component, and molecular function. So just click here, the biological process. I think we do not have sufficient data for this gene. If you are searching some another gene, you will have something here. So this is just a demo. So let me change it my BMP4. Maybe we have some data then. So let's say BMP4. So in case of BMP4 we have this. Now you can see the gene ontology. The biological process is too many biological processes. If you want to read about, let's say you want to read about the osteoblast differentiation, just right click new and it will give you some further details how this gene is working in the biological process of osteoblast differentiation. The same cellular component and molecular function, it is involved in cytokine activity or protein binding or growth factor, whatever. You can find more information from here. And this will be the gene summary. We have changed our genes to BMP4, so you can see this is the gene ID, the gene description. If you want to see this gene in UniProt for the sequence information, right click here and now it's open the BMP4. We can see it in the UniProt. If you need the sequence, just click here. The sequence information is here, and we have already used this database in one of our a video before in the modeling, like how to copy the sequence, blah blah. And if you are searching for the structure information, whether it is solved in the PDB database or not, click here. And then let's see, we do not have a structure yet, so we can only see the AlphaFold predicted structure. So once again, okay, coming back to this section, now we are moving to the single cell transcriptomics, the part 2. So here this is the gene. Now you can type wait, okay, you can type the gene name, let's say BMP4, okay, and search for the single cell transcriptomic analysis. And you can see here these are all the log, FC, average log, and the p-value, whether the data is significant or not, and about the data sets type, etc, etc. So you can also take much more information from here. Of course it will need a lot of time to explain each and every section of this database. And the next one is epigenomics. For example, so when you click the epigenomics, it tells you that we have 15 data types and 12 of them is from the chip assay and 3 are from the sequencing, and then the regenerative models are here. So what you need, let's say we just click here search first. When you click here search, you have these data sets. You can see all of them are from the histone modifications. It will be different for your gene or for your you may have less or more data set. So now the beauty of this database is you can do research, or you can find out some interesting thing using these data sets. These data sets are actually taken from the Geo database. So let's say I click it here that I'm interested in this data set. So now this data set is selected here. Now which gene you want to search in this data set. Let's say I'm interested in BMP4. Now go to this section. It's a genome browser, and let's say what it shows us. Okay, this is about this gene and some information. You will need to study a little bit about the data sets. Now if you want to study how to work here, you need to study this help section. Further, you can use their maps, their apps for the gene plot, scatter plot, session, screenshot. So just click here, gene plot, select your data set, add, and you will have some kind of gene plot or gene data set, etc., etc. So you will have to select a gene, each and everything. So just keep in mind. Now the next section is pharmacogenomics. This is an interesting section, and I know most of you will take interest in this section. Why? Because it tells you about the compounds which were tested in different assays, and so here you will need to search the compounds. And for example, if you don't know the compounds, then you can just type your gene name. Let's say BMP4 is my gene, so just select the gene. Now here we have one entry, and that is dibenzyl. So this is the treatment, or this is the compound, and this is the DUI. You can this is the DUI of that paper, so you can read it how it works. So the same way you can search whatever compounds, all species. This is in mus molluscus, and this is the one we search it. So it's up to you what you search. How about your gene? Let me search. A gene comes in my mind, HER2, ERB2, ERP. Oh, I think BRCA1. Okay, because these are not regenerative genes, so that's okay. I was just confirming that if you are not getting entry for any gene, it means that gene is not involved in the regenerative biology. So you will get details on each and everything for those genes which are involved in a regenerative biology or regenerative process. Okay, so the question is, let's say BMP4 is your gene, and you found it that dibenzazepine is a compound that is involved in some kind of inhibition, or in some kind of or simply we can say that this compound is involved as antagonist or agonist. So now what we can do further. So if you remember, I have a video on the YouTube channel, and the video title is how to mine public database for similar compound. So for example, from here you found this compound works in that gene, works as an antagonist or works as an agonist for that gene. Then I would suggest, if you are interested in pharmacogenomics, I would suggest just follow this video and collect a library of certain compounds, 1000, 100, 500, 50, 60, whatever do you like, from the PubChem database, and dock all that library, which we call it virtual screening, and you will find, if you find the bitter compound, then 1WR1, then you can report that compound accordingly. So it will be an interesting area for you if you want to work in regenerative biology. And finally, you can work for metabolomics. I think people may not be very take a lot of interest in metabolomics, because people are those who are taking in interest in metabolomics, most of them are linked with studies like LCMS or GCMS. So if you have those luxury of LCMS and GCMS, then don't go from this angle, come from the experimental angle and bring your metabolomics data, and we or other people can help you how to run your bioinformatics pipeline on it. So to conclude this video, this is a database. You can use it for regenerative projects or regenerative study, and you can find out genes involved in regenerative biology for a specific organs, their transcriptomic data, their single cell transcriptomics, their epigenomics, their pharmacogenomics, their metabolomics, and you can also explore this help section further. Stay tuned to the channel. In near future, I'm going to teach you many smaller interesting things which will help you in combining these smaller videos, will help you to in your project, in any kind of project. So thanks for now, and I hope this video help you, and stay tuned. If you have any question, please ask me. If you like my videos, please subscribe this channel. Thank you. Bye bye.